Now let's talk about the blood clot formation. So we go from a plug, which is a temporary hemostatic mechanism, so temporary blockage of the site of injury, and then we're going to convert that into a more secure, more permanent blood clot. So clotting is coagulation. It's where we convert the blood into a solid gel-like structure that will create a clot or a thrombus and actually heal the site of injury. This occurs around the temporary platelet plug, and it's a hemostatic defense mechanism. So again, it's going to stop the bleeding and encourage the healing at the site of injury. The final steps in the blood clot formation include fibrinogen, which is converted into fibrin, the active form. This creates a loose, sort of disorganized, again, somewhat temporary fibrin. And then that can be converted into fibrin, which is a more permanent, secure fibrin clot or blood clot. So we go from a loose fibrin clot to a more permanent fibrin mesh or blood clot. And this is what that looks like, right? We have these blood cells being trapped in that mesh. It's kind of like a net initially, but then it becomes a more secure film or a clot or plug temporarily later on. Or sorry, permanently later on. So let's talk about the steps in this cascade, this blood clotting cascade. And there are two pathways here. There's an intrinsic pathway that's kind of more important. And then there's an extrinsic pathway that feeds into the intrinsic pathway. So the steps that you need to know for your purposes are the final steps of the intrinsic pathway, which are highlighted here along the center. So we go from factor 10A to thrombin, right? 10A actually helps to activate prothrombin or factor 2 to thrombin, factor 2A. And what is required for that is factor 5, calcium, and platelet factor 3 or PF3. So we go from 10A, and 10A helps to convert prothrombin to thrombin. Thrombin is very important. Thrombin does two things. It can activate the inactive factor 13 to 13A. It also activates fibrinogen, the inactive form, to fibrin to create the initial fibrin or loose meshwork of the clot. So thrombin, very important in the securing of that initial fibrin clot. Now, factor 13A will then convert the loose meshwork, fibrin, into a more stabilized meshwork, which is the final fibrin clot. Okay, so I'll go over that here again. We have 10A with the help of factor 5, calcium, and platelet factor 3. That is being converted from prothrombin, or that is helping prothrombin be converted into thrombin. Thrombin, very crucial. It does two things, converts inactive factor 13 to factor 13A, and then factor 13A turns the loose fibrin into a stabilized fibrin. And then thrombin can also activate fibrinogen, which is inactive, into fibrin to make that loose meshwork. All right, now the other pathway here is the extrinsic pathway. And then we're also kind of zooming out to look at the other parts of the intrinsic pathway that are further upstream. Now, you don't need to memorize these. You don't need to know these in depth. I just need you to know that they exist. So the details of the extrinsic pathway here that feeds into the intrinsic pathway, specifically at the conversion of factor 10 into 10A. This is where the extrinsic pathway meets the intrinsic pathway. And then further upstream in the intrinsic pathway, we have factor 12, 11, 9, and then ultimately at factor 10A is where both pathways combine. So what you kind of need to know is where both of the pathways meet at factor 10A, and then the way that prothrombin gets converted with the help of 10A and these other factors to bring about the other steps that we described. So from factor 10A onward is what your focus should be. All right. Let's talk about some of the clotting factors and how they get activated here. So the clotting factors are made by the liver. They are then secreted into their blood. Now they need to be in their inactive form. Otherwise, all of our blood would clot, right? All of our blood in our vessels would clot if these factors were secreted into the blood in their active form, but they're not, right? And so they become activated in this cascade that we just described. So that cascade is very important into ensuring that we only clot areas that are needed. Otherwise, we could end up having blood clots. There are diseases that have excess blood clot formation that can lead to thrombi and emboli and strokes and pulmonary embolisms because of inappropriate clotting of the blood. So having this cascade where one factor activates the next, is sort of a mechanism to make sure that clotting is intentional and only the areas that need to have that clot are going to see these clotting factors activated. Now, if we took all the clotting factors out of our blood, that would be serum. So serum is what's left behind when we take all of these clotting factors out. And then we need other substances to help with clot formation as well. So calcium, which is also called factor 4, platelet factor 3, which is another component that is needed in that activation of thrombin, right, from prothrombin to thrombin. Now, what are the factors that limit clot formation? These are called anticoagulants. And anticoagulants are proteins. They're also circulating in our blood, just not activated. And they're also found on the surface of our endothelial cells. This includes tissue factor pathway inhibitor, TFPI, and that is secreted again by healthy cells to inhibit the extrinsic pathway. We also have thrombomodulin, which is secreted by healthy endothelial cells, and that can bind to thrombin, forming a complex that activates protein C. So when thrombomodulin binds to thrombin, it kind of deactivates thrombin, but it makes another complex that activates protein C, and protein C is itself an anticoagulant. It's going to prevent clotting or clot formation. So two things we want to kind of keep separate here. There are two factors that prevent platelet aggregation, right? That is prostacyclin and nitric oxide. And those are secreted by healthy endothelial cells. And then there are two factors that help to prevent clot formation, and those are tissue factor pathway inhibitor and thrombomodulin, which prevent the blood clot formation, right? They help to prevent clotting or anticoagulants. So two different things that are secreted by healthy cells that prevent platelet aggregation and that prevent platelet or blood clot formation. Now, we also have factors or products that can dissolve a formed clot. These are called plasminogen activators. And plasminogen is basically what converts, or plasminogen activators convert plasminogen to plasmin, like plasminogen is inactive, plasmin is the activated form. Notice how all of these things need to be activated, right? They're all in the blood. And so we don't need to have them activated as they're circulating in the blood. They're activated only as needed by various feedback mechanisms and various activators. So plasminogen converted to the active form plasmin. Plasmin is an enzymatic product that can actually break down, degrade, or digest the fibrin clot once it's formed, all right? An example of plasminogen activators is tissue plasminogen activator, TPA, and TPA made by healthy endothelial cells during clot formation can actually be destroyed, can actually help to destroy the fibrin, and it is secreted and activated by fibrin. So when there's excess fibrin formation, lots of fibrin clots, that can trigger the formation of TPA by the healthy endothelial cells, and that can help to degrade excess fibrin after that after that site of injury has been healed. All right, so TPA is an example of a plasminogen activator. which helps to create plasmin and then break down that fibrin clot. Now, the role in terms of these coagulation factors is very important. When you look at some of the diseases and disorders that can be brought about, if we have inappropriate clotting or if we have genetic diseases that prevent certain aggregation of platelets or even the clotting factors in the cascade to be produced. So one of those is hemophilia. And hemophilia is a genetic disorder. It's actually X-linked, so we see this more commonly in males than in females because it's X-linked. And it's a disease where we have deficiency in the genes that produce coagulation factors. So there's different types of hemophilia. Hemophilia A is a deficiency type in clotting factor 8. Hemophilia B is clotting factor 9. But different factors can be deficient, and that can lead to bleeding disorders, and that can be genetically passed on. Another example of this one, von Willebrand disease. This is a deficiency in von Willebrand factor. So this will have an effect on platelet adhesion, which is the initial part of that platelet plug formation. And so that can also present if patients have had easy bruising, easy bleeding, bleeding gums, bleeding joints, bleeding in their muscles with very minimal injury. This can be very, very dangerous. They can be bleeding, bleeding out, which can lead to death or very severe conditions because of this easy, bruising or bleeding. And then vitamin K deficiencies. Vitamin K itself is very important as a precursor to making these cofactors, and it is itself a clotting factor. So decreased synthesis of clotting factors can be produced if we have a deficiency in vitamin K, right? One of our fat-soluble vitamins. So these are some of the clinical connections to the importance of blood clotting and the role of the coagulation factors. Now, in terms of aspirin, aspirin can also be an anticoagulant, but only at low doses. It actually can inhibit thromboxane A2. Remember, thromboxane A2 is made by platelets. So it's one of those things, along with ADP, that is secreted by platelets when they first begin to aggregate, that can sequester or aggregate more platelets to the site of injury. And so if we have inhibition of thromboxane, that can stop platelet formation, platelet aggregation at the site of injury, which can be an anticoagulant. Now, this is only at low doses, right? This is where we have baby aspirin. If you've ever heard of baby aspirin being given to patients as a heart protectant, it protects against heart disease, protects against strokes, right? This is why it protects against strokes. It prevents any minimal clot formation, any atherosclerosis, blockage of our heart vessels, blockage of our brain vessels because of that coagulation and clot formation. So baby aspirin, low doses, less than 81 milligrams or so, is very important as an anticoagulant. While at high doses, it actually promotes platelet aggregation because it inhibits prostacyclin. And prostacyclin is one of those factors that is made by healthy endothelial cells that blocks platelet aggregation. So at high doses, it stops it inhibits the inhibitor of platelet aggregation, which means that it promotes platelet aggregation or platelet clotting. So high doses is procoagulant, low doses is anticoagulant.